The Krebs cycle, also known as the citric acid or TCA cycle, is the third stage in aerobic respiration, after glycolysis, which takes place in the cytosol, where glucose is broken down into two pyruvates, and the link reaction, where pyruvates are converted to acetyl-CoA and enter the mitochondrion. This short bioanimation will visualize the Krebs cycle and demonstrate how NADH and FADH2 are produced within the mitochondrial matrix. The citric acid cycle initiates with the final product of the cycle, oxaloacetate and acetyl-CoA. Citrate synthase catalyzes the condensation reaction of acetyl-CoA and oxaloacetate, and a 2-carbon acetyl group is transferred to the 4-carbon acceptor compound, forming citrate. Next, citrate is isomerized by the enzyme aconitase, which facilitates the hydroxyl group migration to form isocitrate. Now, oxidative decarboxylation of isocitrate to alpha-ketoglutarate is catalyzed by NAD+ dependent isocitrate dehydrogenase, producing CO2, NADH, and H+. In the next step, alpha-ketoglutarate dehydrogenase catalyzes the conversion of alpha-ketoglutarate to succinyl-CoA. This irreversible stage generates NADH, CO2, and a H+. This division releases free energy, which is utilized to attach a phosphoryl group onto a guanosine diphosphate molecule, forming guanosine triphosphate. This is catalyzed by succinyl-CoA synthetase. The GTP is used to form ATP molecules, with the reaction ADP plus GTP plus nucleoside diphosphokinase goes to ATP plus GDP. Now, an oxidation reduction reaction occurs, where the enzyme succinate dehydrogenase makes possible the oxidation of succinate to fumarate, abstracting a pair of electrons and 2 H+ ions, and producing a reduced FADH2 from FAD+. Next, the hydration of fumarate to the L-isomer of malate is catalyzed by fumarase in the penultimate stage of the cycle. In the final stage of the Krebs cycle, malate dehydrogenase is the catalyst in the oxidation of malate to oxaloacetate and transfers the electrons onto NADH. This process is highly endergonic and must be balanced out with other exergonic steps. The final products in the first turn of the cycle are 1 GTP, 3 NADH, 1 FADH2, and 2 CO2. As 2 acetyl-CoA molecules are produced from each glucose molecule during glycolysis, the Krebs cycle has to occur twice, creating 2 GTP molecules, 4 CO2 molecules, 6 NADH molecules, and 2 FADH2, the latter two of which will be crucial to the fourth and final stage of aerobic respiration, the electron transport chain, where they will facilitate the generation of ATP from ADP. Thank you for watching. This video was created by Paige Williams, Kev Tomlinson, Patsy Young, Clarissa Potts,